So we're on to our next part of the brain. So here we are at the brainstem. And if you remember, let's kind of keep a running list here of our brain regions. So first would be cerebral hemispheres. Right? The second region of the brain was the diencephalon. And then now we're on to the third region, which is the brainstem. And so we're basically just kind of heading down. So this area through here was our cerebral hemispheres, or cerebrum. And then we had this area kind of through here, which was the diencephalon. And now we're down a little further. So this is going to be brainstem. And you can see this on this image here. It's going to be this area, just inferior to the diencephalon. So here's brainstem. And we can take the brainstem and we can divide it up into different sections. So we're going to have the midbrain, the pons, and then our third region, which is going to be on the next slide, is the medulla oblongata. So what we see with the midbrain are structures that are included in the corpora quadrigemina. And so this is going to be this tiny little area right here. It's shown in orange on this image. And this consists of structures called the superior and inferior colliculus. And so the way this looks, in cross-section, you're basically going to see these two little bumps in this area. If we were to keep the brain whole and not cut it, a sagittal plane cut like we see here, if we just look at this section from the back, we're going to see actually these four little round bumps that look kind of like this. And these are going to be the colliculi. So the ones on top are going to be the superior colliculus or colliculi, and the one on the bottom will be the inferior. So superior colliculus is known for visual reflexes. So this is how we're able to coordinate our head and eye movements when we're visually tracking objects. I give the example of going to a tennis match and you're watching the ball go back and forth, or something catches your eye and you visually track it and coordinate movements of your eye as well as movements of your head. The inferior colliculus is going to be our auditory reflex center. So this is the area where auditory information is going to come into the inferior colliculus and it's going to be relayed to the sensory cortex, and namely the auditory cortex of the temporal lobe. This is also the location of our startle reflex. So let's say you're walking through a parking lot at night and you hear a door slam or something startles you that you're not expecting and you jump, there's your startle reflex. And that is also being relayed through that inferior colliculus. The second structure that's related to the brainstem is the pons. And that's going to be this structure here. So it's a little bit rounded anteriorly, and it's going to be just inferior to the midbrain. So here's our pons, and pons means bridge. And so that's exactly what we see here, is it's basically a relay station between the brain, which is going to be above it, and then the spinal cord, which is going to be down here leading up into it. So pons is basically a bridge that controls information traveling from lower centers up to higher centers. And then our third section of the brainstem is going to be the medulla oblongata. And a couple things happen here. Number one, this is where we have crossing of our spinal tracts. So if you see here, this is a frontal plane view of the brain, and we've got tracts that are originating in the motor cortex and the sensory cortex, all throughout our higher cortical areas of the cerebral hemispheres, and they're going to travel down. And then if you'll notice in this area, a lot of the fibers are going to cross to the other side and continue traveling down the spinal cord. Well, this is called the decussation of the pyramids, and this particular area of crossing is located in the medulla, And this is the reason why the right side of the brain controls the left side of the body, because we have these fibers that are crossing here. Other functions of the medulla are to maintain homeostasis. It plays a large role in this. So we have cardiac center located in the medulla, and this is going to control heart rate and strength of contraction of the heart. We have the vasomotor center, which is going to control vasodilation, vasoconstriction of blood vessels, and therefore blood pressure. We have the respiratory center, which is going to control rate of respiration as well as depth of respiration. And then the medulla also controls vomiting, coughing, and swallowing. Okay, so now here's our fourth region of the brain. We're on to cerebellum. And cerebellum is located underneath the occipital lobes. I'm going to back up to this one here. Here's our cerebellum here. Okay, so it's underneath the cerebral hemispheres. This is the cerebellum. And basically what we have is, if you look on the inside of the cerebellum, it's got these branching kind of arrangement to its tissue. Well, this is called arbor vitae, and that means tree of life. Okay, and so arbor vitae is the particular branching that we have within the cerebellum. Now, cerebellum does also have a cortex, a cerebellar cortex, which is going to be the outer part of tissue. And then it's got cerebellar white matter. Just like the cerebral hemispheres does. So that white matter is also present in the cerebellum. And the way that the cerebellum is arranged is that it's got two lobes. So here's our cerebellum here, and this is looking at it from the back, and you can see here's one lobe of the cerebellum here, and then here's the other lobe on this side. Now, this area in between the cerebellum is called the vermis, and it basically connects the two lobes of the cerebellum. Cerebellum is also mapped out, it has a mapping of our body on it, and we call that the cerebellar homunculus, similar to what we saw in the primary motor and primary somatosensory cortex, or cortices. So what's the function of the cerebellum? What does it do for us? Well, it basically plays a role in smooth, coordinated movement patterns. So it's not really just balance necessarily. It's really doing coordination. And so one of the ways that we can test this clinically is performing what we call the finger to nose test. And in this type of test, I would hold out my finger and I would have my patient take their finger, tap it to my finger, and then tap their nose. So in this way, they're able to gauge their movement as well as stopping that movement at an appropriate time, so they don't overshoot my finger and that sort of thing. And one of the pathologies associated with the cerebellum is called dysmetria. And that's what happens when patients don't do well on this finger to nose test. When they're not able to gauge distances, it results in clumsy movements. And that's what we see. Patients sometimes will have stroke involving the cerebellum, and this is what happens. So I've included a couple videos for you to take a look at. So make sure you look at those. It's interesting to see patients walking who've had cerebellar strokes, because it is very clumsy. I also want to note too that the cerebellum is located underneath the occipital lobe. And if you recall, this is going to be important in just a few minutes, this fissure that separates the cerebellum from the occipital lobe was called the transverse cerebral fissure, and that was that deep groove between the structures. So that's going to be this groove here. Okay, and we're going to come back to that in just a few minutes. Okay, now we also have some examples of what we call functional brain systems in the central nervous system, and I've picked out two of these just to talk about briefly. One of them is the limbic system, and limbic system deals with fear. So this is our fear response. Important structures that are part of the limbic system are the amygdala, which is going to be this small structure right here. Also, hippocampus, which plays a large role in memory, and then hypothalamus. So we have a structure called the fornix that connects all of these structures together, and it basically runs just superior, kind of arches superiorly over top of the thalamus. And the limbic system is active in dealing with fear and the emotion of fear, the memory of fear. It's going to compare it to situations that we've been in before. And then the amygdala is basically our emotional response to whatever situation we're in. We also pull in our prefrontal cortex, where we have higher cognitive functions that tell us kind of rationally how to react. But they also are receiving input from the amygdala, which is going to be more of the emotional response. The second system is going to be the reticular formation, and this basically has to deal with arousal. And so this is a group of neurons that are located along the length of the brainstem. So we're going to find this here. And basically they receive impulses from the body. So we're receiving sensory information from the body that's being relayed up through the reticular activating system, and it's relaying that information up to the cerebral cortex. And so as a result, this is what keeps our cerebral cortex alert and conscious. And this is the reason why some people, if you have friends or know other students who can study in very busy like coffee shops, they tend to like that because of the arousal. It kind of maintains this level of arousal of their cerebral cortex. Other people would just find it distracting. But that is a function of that reticular formation. We see that the reticular activating system is inhibited by sleep centers in the hypothalamus. So we basically have to shut it off in order to go to sleep. Alcohol will inhibit it as well, and so will tranquilizers. Okay, so we're on to meninges. And meninges are basically these connective tissue membranes that cover the brain and spinal cord. And they're very protective, especially the outermost layer. It's very thick, so it offers a lot of protection. It's going to contain blood vessels that supply the brain. It also contains cerebrospinal fluid to keep our brain buoyant and protected. And it's also going to form partitions in the skull. So it kind of goes in between the brain and anchors to the skull itself so that your brain doesn't go all sloshing around in your skull when you're moving around or playing sports or that sort of thing. So we have 3 layers of meninges. The outermost layer, so this is going to be the most external layer, and this is going to be the dura mater. And so here's our dura mater here. So if we basically peel down just underneath the bone of the skull, we're going to see the dura mater. So this is going to be most external. Okay, and this is basically a double-layered fibrous connective tissue. The outer layer is the periosteal layer, and it's attached to the bone underneath the skull. The deeper layer is called the meningeal layer. Okay, so periosteal and meningeal. And these layers actually separate in certain areas of the brain to form the dural venous sinuses. And this is where we have venous blood draining from the brain into the venous sinuses, and then they're going to travel down through the internal jugular, back to the heart. So we have certain areas of the dura mater that are kind of folded and are located in fissures that we find in the brain. Okay, and we call these particular ones partitions. So that's kind of an important word because you're going to see that written on tests and that sort of thing. So these are going to be specific partitions of the dura mater. And we've got 3 of them. Okay, and the first one is going to be the false cerebri, and that's located in the longitudinal fissure in between the left and right hemispheres. The second one is the false cerebelli, and that's going to be located in the vermis of the cerebellum. And if you remember, that separates the cerebellum into 2 lobes. And then the third one is going to be the tentorium cerebelli, and that's located between the cerebrum and cerebellum in that transverse cerebral fissure. So certainly knowing all 3 of these partitions and where they're located would be very important. Underneath the dura mater, we're going to have a space, and we call that the subdural space. And this is going to contain cerebrospinal fluid. Let's go back to this. So here, where does the false cerebri attach anteriorly? Well, it's going to attach anteriorly to the crista galli of the ethmoid bone. It's that ridge of bone that we learned about in the skull. And false cerebri is going to come along and be anchored to that ridge of bone. Our second layer, so arachnoid mater, is going to be our middle layer. Okay, and this is more of a loose covering. It's not quite as thick and dense as the dura mater, but it does have a it does cover the brain. And above it, we've got the subdural space, which is going to be in between dura mater and arachnoid mater. And that contains cerebrospinal fluid, like we talked about. And then underneath it, we've got pia mater, which is going to be our deeper layer. But arachnoid mater is basically going to be our middle layer of meninge underneath the dura mater. Now, this is our most internal layer. This is pia mater, and this is this stands for gentle mother. So pia mater is really interesting. I always tell students it's kind of like saran wrap on the brain. It's very thin and delicate. Dura mater is really tough and thick. All my students are surprised at how tough it is. Pia is very thin. It's like a very thin saran wrap. And it actually is going to travel into all of the sulci and convolutions in the brain. So where dura and pia basically cover the brain just like a blanket, dura and arachnoid cover the brain like a blanket, pia mater is actually going to dive down and travel into the sulci. Okay, it's a very delicate connective tissue, and it's highly vascular. It's got a lot of blood vessels that are located in it. Okay, so this is what you can see from the in a microscopic image. Look how nice and thick that dura mater is. And then we're going to have a subdural space with arachnoid mater is going to be our next layer. And then we're going to have a subarachnoid space with pia mater being our next layer. Okay, and so this brings us to our ventricles. And ventricles are going to be hollow chambers that are found within the brain and spinal cord. And they're filled with cerebrospinal fluid that are being produced by this little tangled knot of capillaries located within the ventricles called the choroid plexus. Okay, so these chambers, these hollow chambers, are filled with cerebrospinal fluid, and they're basically continuous with each other. So we see this flow of cerebrospinal fluid throughout the ventricles. So we've got two lateral ventricles. So we're going to have a left lateral ventricle that's located in the left cerebral hemisphere and a right that's located in the right cerebral hemisphere. And you can see here, we're looking at the brain from the front. This area here, this would be my left lateral ventricle. On this side, this would be my right lateral ventricle. They kind of look like ram's horns. The third ventricle is going to be in this area here, and it basically circles around the thalamus. So if you picture where the thalamus is, that's where the third ventricle would be. And the fourth ventricle is going to be located anterior to the pons. And it's going to be like this little kind of triangular shaped structure. And so if we look here, this is our fourth ventricle here. Okay, so we have a couple things. The cerebral aqueduct is going to be this connection here, this little passageway that's connecting our third and our fourth ventricles. Okay, so this little passageway here is going to be cerebral aqueduct heading down to fourth ventricle, and then the fourth ventricle is going to continue all the way down through the spinal cord as the central canal in the spinal cord. Okay, so we're basically going to see this flow of fluid throughout all of those ventricles. Some histology with the ventricles, what we see is that ventricles are lined with a specific type of cell called ependymal cells. Okay, and ependymal cells are important because they're joined with tight junctions. Remember, we had 3 types of cell junctions, and tight junctions were one of them. And basically, the point of having a tight junction is to be able to have a lot of control. So that's exactly what we have here, because we want to have control over what has access to our brain tissue and brain circulation and what doesn't. So the ependymal cells that line the ventricles are going to be joined together by these tight junctions, and therefore they're able to control flow going from the capillaries into the ventricles. Okay. So the choroid plexus, this is how the cerebrospinal fluid is made. The choroid plexus is a knot of capillaries that's found in all of the ventricles. So all ventricles have choroid plexus in them. Therefore, they all make cerebrospinal fluid. Okay, all your ventricles actually make cerebrospinal fluid. And what we see is we have the choroid plexus that comes down this little knot of capillaries, and it's surrounded by the ependymal cells. Okay, and so that way we are able to control flow. So what we have is basically a filtrate that's going to travel from the capillary and it's going to become cerebrospinal fluid. So cerebrospinal fluid forms as a filtrate from the capillary bed, traveling through those ependymal cells into the ventricles. Okay, and this is how you can see the flow of cerebrospinal fluid throughout all of those, all of the ventricles, and even around the brain, and it's basically bathed in cerebrospinal fluid. And what this does is it forms a cushion. So it makes our brain buoyant. Okay, it adds some buoyancy. It reduces our brain weight by 97%. Otherwise, like a brain would kind of collapse upon itself with its weight. And basically, cerebrospinal fluid is very similar to plasma, a little less calcium and potassium included in that. It is interesting to note, we talked about this, choroid plexuses are located within each ventricle, therefore each ventricle plays a role in the production of cerebrospinal fluid. We have ion pumps that regulate what is able to flow from the capillary into the cerebrospinal fluid, and that's related to the tight junctions between the ependymal cells. And then there are also cilia on the ependymal cells, which I think that we saw here. You can see there's cilia lining the ventricles, which is able to help cerebrospinal fluid flow from one area to the next. So that's what we see as well. This is a close-up of choroid plexus in the lateral ventricle of a real brain, and that's what it looks like. You see a little tangled knot, and that would be a choroid plexus. And you can also see it in this image too, these red structures located in each ventricle would be the choroid plexus. Okay, which brings us to our blood-brain barrier. And that's basically what we call the barrier that we have between the endothelial cells of the capillaries in the brain. They have tight junctions, so we have lots of control over what flows in and out. And astrocytes, which are a type of neuroglial cell in the central nervous system, are the cell types that stimulate those endothelial cells in the capillaries to form the tight junctions. This is very protective. This allows us to control what has access to our brain. And what we see is that lipid-soluble substances are able to pass very freely. Otherwise, for other substances, we need specific modes of transportation in order for that to happen. So this is what we see when we compare a capillary in the brain, which is formed as a result of that blood-brain barrier. This would be just a capillary in general elsewhere in the body. And you can see these fenestrations or these pores in the capillary in other locations in the body. Well, what happens in the central nervous system is that astrocytes, which are a supporting neuroglial cell, stimulate the endothelial cells of the capillary bed to form tight junctions. So we basically have no pores in the capillaries of the brain. So we don't get quite as much movement in and out. The blood-brain barrier is typically uniform throughout the central nervous system, except in one key area, and this is the vomiting center of the medulla. And what happens here is that the blood-brain barrier is not nearly as intact, so that we are able to monitor our blood for poisonous substances. We have chemoreceptors that are located in the medulla, and they help monitor our blood for poisonous substances. If something is detected, it's going to stimulate the stomach, and it's going to activate our vomiting reflexes.